So how do you figure out what's actually true versus what just feels true? I mean, how do we really know what's real? In a world that's just overflowing with information, that question is more important than ever. Well, the answer lies in this really powerful process for understanding reality. Okay, let's dive into this.
To really get this, we've got to go way back in time. For thousands of years, humanity was just plagued by these deadly diseases. We're talking cholera, the Black Death. They were terrifying, they were completely mysterious, and people were desperate to know what was causing them. So what do you think they blamed it on?
Get this, for more than 1,500 years, the smartest people in medicine, from doctors in ancient Rome all the way to Victorian London, they all agreed on one thing. Disease was caused by bad air, some kind of poisonous, foul-smelling fog.
Yeah, it had a fancy name, the miasma theory. It was first dreamed up by the Greek physician Galen way back around 200 BCE. The idea was that diseases were spread by miasma, this toxic mist that rose up from rotting stuff. Basically, if it smelled awful, it was probably going to kill you. It sounds kind of strange to us now, but for centuries that was the scientific consensus.
And look, we're not just talking about some fad that came and went. Miasma theory was the bedrock of medicine for a mind-boggling amount of time.
Okay, take a look at this timeline. This really puts it into perspective, right? From Galen's idea all the way into the 1800s, miasma theory was king. The concepts that would eventually grow into germ theory, they were just tiny little blips on the radar for most of that history.
So that's the big question, isn't it? How on earth does a better, more correct idea take down a giant that's been accepted as fact for over a millennium? It takes a total shift, a completely new way of thinking.
So how do we make that massive leap from bad air to germs? And here's the thing. It wasn't some sudden aha moment. It was a slow, deliberate revolution in thinking.
It was more like a slow burn revolution, started by a few key people who planted the seeds of a new idea. First, back in the 1500s, this guy, Fracastoro, suggested that maybe tiny, invisible, seed-like things were the real problem. Then, over 100 years later, a lens maker named Van Leeuwenhoek built a killer microscope and was the first human to actually see these little animalcules, what we now know are bacteria. But the idea didn't really catch on until Louis Pasteur came along in the late 1800s and delivered the knockout punch with undeniable proof.
Now, these folks, Pasteur, Leeuwenhoek, and the others, they weren't just throwing darts in the dark. They were using a structured way to ask questions and find evidence. You can think of it like a detective's toolkit for solving the mysteries of the universe. And that toolkit? Well, we call it the scientific method. It's a pretty logical process, really. You see something, you form a possible explanation, that's your hypothesis. Then you use that to make a prediction you can test. You run an experiment, and finally, you look at what you found and tell people about it.
And it really does all kick off with that first super important step, just noticing something, seeing a pattern or a puzzle in the world that just doesn't quite make sense.
Okay, check out this amazing story of a doctor named Ignaz Semmelweis back in the 1840s. He noticed something absolutely terrifying in his hospital. In the ward where medical students were delivering babies right after they'd been doing autopsies, mothers were dying from this thing called childbed fever, a lot of them. But in the places run by midwives, or for women giving birth at home, it was way less common. His hypothesis? The doctors were carrying cadaverous particles, as he called them, on their unwashed hands. And his solution, which was considered totally radical at the time, just wash your hands.
Okay, so a hypothesis is great, right? Even a brilliant one like that. But it's just an idea. It's nothing until you can actually test it. This is where science stops being about just thinking and starts being about doing.
And the absolute best way to do that is with what's called a controlled experiment. The whole point is to change just one thing, that's your independent variable, and see how it affects an outcome, the dependent variable, all while you keep everything else exactly the same.
Let's break it down with a classic example, how Alexander Fleming discovered penicillin. He had petri dishes full of bacteria. Some were near an open window, some weren't. The ones by the window, that's the test group, they got some mold spores in them. That was the independent variable. The dependent variable was what happened to the bacteria, and what he saw was amazing. Where the mold grew, the bacteria died. The dishes away from the window, the control group, the bacteria in them were just fine. Cause and effect, right there in front of him.
But there's a problem, right? We're all human. Even scientists can kind of hope for a certain result, and that can unconsciously influence things.
To fight that, they came up with another crucial tool, the blind experiment.
So in a blind experiment, the participants have no idea if they're getting the real deal or a fake one, a placebo. This was famously used in the huge Salk polio vaccine trials. But to take it up a notch, you do a double-blind study. This is the big one. Here, neither the patients nor the researchers handing out the stuff know who's getting what until the whole thing is over. It strips out bias from both sides. It's the gold standard.
Okay, so we've talked about all these scientific tools, controlled experiments, blinding, but why should you care about any of this? Because knowing this stuff is like having your own personal baloney detector for all the information that gets thrown at you every single day.
All right, one of the biggest, most common mistakes people make is mixing up correlation and causation. Just because two things happen around the same time doesn't mean one caused the other. Like ice cream sales and shark attacks both shoot up in the summer. Does eating ice cream cause shark attacks? No, obviously not. There's another factor, the warm weather, that's causing both. We hear this kind of thing all the time, right? And it sounds super compelling.
And that sounds awesome, but it's just one story. In the world of science, that's called anecdotal data, and it is not a real experiment. We have no idea what else was going on. Was your cousin also starting a new workout plan, drinking more water? We don't know what actually caused the weight loss. For a result to be scientific, it has to be replicable. You have to be able to get the same result over and over again under controlled conditions. One good story is not proof.
So let's put together your scientific literacy toolkit. A few simple things to keep in mind. First up, watch out for those anecdotes we just talked about. Always ask, is this just one person's story or is it based on a real controlled study? And if you can, try to find the original study. Don't just read the clickbait headline about it. When you look at it, pay attention to the graphs and charts. They often tell a much clearer story than the summary does.
And this all leads to probably the most important takeaway here. Science isn't just a big pile of facts that are set in stone. It's a process. It's a way of asking questions, of testing and retesting. It's the best tool we've ever come up with for getting closer to what's real, even if it means we have to admit we were wrong for, 1,500 years.
So I'll leave you with this one last thought. We finally moved past bad air. So what's something we all believe is true today that future generations will look back on and just shake their heads? What's our miasma theory? Something to think about.